So here we are going to discuss about mechanism of activation of T cells. How T cells are activated. So first of all, here I am going to tell you about T cells work separately. Okay, but when T cells will activate, then B cell will activate. Otherwise, B cell will not activate. Means T cells will activate B cells. Okay, so moving towards the mechanism. So here you can see I have drawn one macrophage. Here we have macrophage. Okay, right here. Now what here will happen? Here it will perform the means perform phagocytes. Phagocytosis. Okay, so when they will perform the phagocytosis, so they will release one type of chemical, that is interleukin 1. Okay, so interleukin 1 will stimulate the T helper cells. What? Stimulate T helper cells. Okay, so then this will go and attach with the here the antigen came and it break down the antigen in the small pieces and present over here. So now this is anti-presenting cell. Okay, so now what stimulated T helper cell will go? These will go and attach to the antigen. Okay, here it will attached. Okay, so here we have here the self, non-self complex will made when the macrophages present antigen. Here you can see the macrophages is presenting the antigen. This is antigen. So macrophage is presenting here the antigen. So that's why these are the self non-self complex when these will be made when the macrophage will present the antigen. Okay, so now what will happen with the help of receptors? Here T helper cell went and here it will read the antigen, means which type of antigen is present, so that they can make the antibodies easily. After reading that, this it will release an other chemical, means stimulating cell will release an other chemical, that is interleukin 2. Okay, after releasing interleukin 2, here the further division of cytosis cell occur. What further division of cytosis T cells? Okay, so here the stimulated helper cell will also activate the B lymphocytes. Activate B lymphocytes, and it will also proliferate divisions of different T cells. Okay, so here after activating the B lymphocytes, as I told you, it will divide into the two types. First one is plasma B cells, second one is memory B cells. It will report the record which antigen come. Here the antibodies will be made. Okay, so now what, like here it will make the record that in the future if the antigen, again this antigen will come, so this will directly convert into the plasma B cells and then it will make the antibodies. So this antibody now circulation will happen of this in the all over the body. Here now see, we have activate the, like here the cytosine T cell activated. Okay, so now what they will do, they will kill the pathogen. Okay, after helper T cells, like after helper T cells, these cytotoxic T cells will attach to the antigen. Here I am writing, after T helper cells, the T cytosis cells will, we can say, attach to the antigen. Okay, so now what this cytotoxin T cell will do when it will attach the antigen, this will release the toxic or you can say perforance. Okay, so this will release the perforance and the plasma membrane of antigen and make the holes in the plasma membrane of antigen. Like here was the antigen, so in the antigen there is a plasma membrane. So it will make like a cytotoxin T cell will go there, make the holes in the antigen of the plasma membrane. Here you can see this is the plasma membrane of an antigen. So this will T cell will make the holes on the surface. Okay, holes and release the granulozyme. Here is another enzyme, that is granulozyme. It will release. Okay, these holes are known as perforation. Okay, perforation. So now here we have the salt water outside. Salt water. Either the salt water move inside the cell and brush the cell with antigen. Okay, this will be done by the some like in T cells and B lymphocytes. But if this will not happen, so here the enzyme will release, that is with the help of enzyme, writing with the help of enzyme, that is enzyme, so they will apoptosis. Okay, so let me explain you once again what happened here. Like you can see what was there. Here we have the macrophage and the antigen come inside the macrophage. Okay, so macrophage will break down this antigen and represent this antigen on the MSC1. Here the antigen is present, so now this is known as anti-presenting cell. Okay, so when this antigen will be represented by the macrophage, so this is known as self-non-self complex will made. Okay, so here the macrophage perform the phagocytosis. Okay, means break down or eat the antigen. So after phagocytosis, it will release the anti-leukin 1, and anti-leukin 1 will stimulate the T helper cells. Then T helper cell will move to the macrophages or the antigen, and here this stimulating T cell will read like which antigen come here. Okay, after reading this T helper cell will release the anti-leukin 2, which will further divide into cytosis T cells and activate the B lymphocytes. And B lymphocytes are divided into plasma B cells and memory B cells. Memory B cell will make the record, while plasma B cell make the antibodies, and the record will be further further record will be there, and antibodies will be circulation in the blood. Now here further we here the cytosis T cell activate, so they will kill the pathogen. Okay, so there with after T helper cell, like here T helper cell went and read this. After T helper cell the cytotoxic C cell will move there, and what they will attach to the antigen. And after attaching the antigen, they will release some type of toxic or perforance and chemicals. Okay, with that they will make the holes on the plasma membrane of antigen. After making holes on the plasma membrane, after making holes on the plasma membrane, in the outside we have the salt water. So the salt water move inside and this cell will be brushed. This what we have the one condition. First condition, this will happen with the like T cells, B lymphocytes, T memory cells, all will perform this. Okay, but another condition, it will release the with the help of enzyme that is granules, it will be apoptosis, means it will be died, it will kill herself by own. This is the second condition. Okay, hopefully it's clear to you. Bye bye.